In the previous series of videos, I explained how stages 1, 2, and 3 of the pentose phosphate pathway were carried out to produce ribose 5-phosphate and NADPH. What we're going to discuss in this video is how do we control the pentose phosphate pathway. It's possible that NADPH might be needed by the cell and maybe ribose is not. So you should think about what pathways would be required to produce NADPH and not ribose. And the next thing we need to think about is if we consider stages 1, 2, and 3, what pathways should we carry out and which ones should we not carry out? Well, hopefully you remember that in stage 1, in a series of 3 reactions, glucose 6-phosphate was transformed into ribulose 5-phosphate in stage 1 of the pentose phosphate pathway, and one of the carbons was oxidized to CO2, and we obtained 2 molecules of NADPH. In stage 2, the ribulose 5-phosphate was converted to 2 molecules of xylulose 5-phosphate and 1 molecule of ribose 5-phosphate. Again, you would have had to start with 3 molecules right here. And then in the carbon shuffling reactions, we could transform those 5 carbon sugars into fructose 6-phosphate and glyceraldehyde 3-phosphate. If we really want to maximize how much NADPH we can get from one glucose molecule, what we should do then is to take those metabolites, carry out gluconeogenesis to get glucose 6-phosphate, and then go through stage 1, stage 2, and stage 3 again. And what we do then is we take, every time we go through stage 1, we lose 1 carbon as CO2. So essentially what happened is you go through this 6 times so that each carbon is completely oxidized to CO2. And then the net yield is 12 molecules of NADPH. So here's the reaction right here. If I were to take all of those reactions and add them all up, essentially what I'd have to do is I'd need to start with 6 molecules of glucose 6-phosphate. I'd end up with 5 molecules of glucose 6-phosphate. But if you subtracted, well, like I've done down here, essentially what happens is 1 molecule of glucose 6-phosphates, its carbons, the carbons are completely oxidized to CO2. And since every time you oxidize a carbon to CO2, you get 2 molecules of NADPH. So for the complete oxidation of 1 molecule of glucose 6-phosphate, you can obtain 12 NADPH. You won't get any ribose or anything else, but this is what the cell will do if NADPH is needed and the other metabolites are not needed. What we need to remember is that glucose 6-phosphate can enter the pentose phosphate pathway, but we've also discussed how glucose 6-phosphate enters glycolysis to yield ATP. So the cell needs to balance its need for NADPH and its need for ATP. So if NADPH happens to be forming faster than it's needed, either for biosynthetic reactions or for glutathione reduction, what happens is the NADPH will then, by feedback inhibition, inhibit the glucose 6-phosphate dehydrogenase enzyme. Remember, that's the first enzyme that catalyzes the very first reaction of stage 1 of the oxidative branch. And then what happens here is that more glucose 6-phosphate then becomes available for glycolysis. So we just discussed what the cell would do if it needed NADPH and didn't need any ribose. But what if we have the opposite need, that we need ribose and we don't need NADPH? Again, you should think about what pathways might be necessary. So what you would do is, if you start out with glycolysis to give you fructose 6-phosphate and glyceraldehyde 3-phosphate, you could then carry out stage 3 in reverse. So you might have noticed when we were going through stage 3 that all of those reactions were reversible. In this case, you'd get 2 molecules of xylulose 5-phosphate and then 1 molecule of ribose 5-phosphate. And then you would convert these molecules to ribose 5-phosphate, and then that could be used for nucleotide biosynthesis. In this case, we don't carry out stage 1 at all. It doesn't make sense to carry out the oxidative branch if NADPH is not needed. So that's the important reaction right here, is that we're using our glucose 6-phosphate to make as much ribose 5-phosphate as possible. So again, if we were to add up all of those reactions, we would find that the net reaction would be, we would start out with 5 glucose 6-phosphates, or 5 6-carbon sugars for a total of 30 carbons, and the 5 6-carbon sugars would be converted to 6 5-carbon sugars with 1 ATP required for an initial phosphorylation. So what's happened is that all available glucose 6-phosphate has been isomerized to ribose 5-phosphate, and you can use that for nucleic acid synthesis. Now suppose you need NADPH and energy in the form of ATP. Again, think about what pathways you might need. If I show that the glycolysis pathway is linked to the citric acid cycle right here, we can see that what we should do then is to carry out stage 1 to get ribulose 5-phosphate, and then stage 2 to get xylulose 5-phosphate and ribose 5-phosphate, and to carry out stage 3 to get fructose 6-phosphate and glyceraldehyde 3-phosphate. That gives us a link to glycolysis. We can then go forward to convert those compounds to pyruvate. We'll get some ATP and NADH there, but then the pyruvate, as shown over here, can enter, can first be converted to acetyl-CoA and can enter the citric acid cycle, producing NADH and FADH2 as substrates for the electron transport chain and yield more ATP. So we should note that the pentose phosphate pathway depends on cell type in terms of its activity. So in the red blood cell, for example, you would need NADPH to maintain glutathione in the reduced form. That helps maintain the integrity of the membrane. There's a lot of oxygen in the red blood cell because that's where the hemoglobin resides that carries the oxygen. So glutathione is quite necessary. On the other hand, red blood cells don't synthesize nucleic acids, so you wouldn't need any ribose. In the mammary gland, the testes, and the adrenal cortex, NADPH is needed for lipid and steroid synthesis. The liver has to maintain a balance between glycolysis and the pentose phosphate pathway, depending on the needs of the organ in terms of biosynthesis, whether it needs ATP or NADPH or any of those other intermediate carbohydrates. In the muscle, you have glucose 6-phosphate, but nearly all of it goes to glycolysis. So in the muscle, there's very little to no pentose phosphate pathway activity in the muscle. So as a final exercise, I'm going to ask you to draw the products of this reaction. This is a reaction that actually occurs in plants, which is not what we talked about today. But suppose I were to give you dihydroxyacetone phosphate and erythrose 4-phosphate, and I gave you the transaldolase. Use what you've learned about the transaldolase reaction to predict the product. So I would ask you at the conclusion of this presentation to do the chapter 13 problems that are assigned in essential biochemistry. There's no instant notes assignments for this chapter. And here are my references for this presentation.